you very much to the MSIF and to Professor Battaglia and Italian MS Society for giving me the opportunity to be here to present what has been done by us, but also shortly, also because my very good friend Gian Vito is not here, to just touch about what has been done in other fields. So, in other fields of stem cell research, of course. Okay, this was an old slide just to provide you with the current, and this was about 10 years ago, but I would say that this is still the current portfolio for treating multiple sclerosis, which include, I'm talking about stem cells portfolio, of course, which include hematopoietic stem cells, mesenchymal stem cells, neuroprecursor cells, and more recently IPS. And I'm proud to say that thanks to the help of many people, but especially to the Italian MS Society, Italy has been really a pioneer in this field with Genova on these two cells and Milan, San Raffaele in these other two areas. So shortly, I'm just going to go through quickly about what is, since the whole title is Stem Cell Future, but of course I will focus mainly on mesenchymal stem cells. What is hematopoietic stem cell transplantation, which is indeed an intense immune suppression, which leads to the ablation or the partial eradication of potential autoreactive T cells, which are part of the immune repertoire. And upon reinjection of hematopoietic stem cells, autologous hematopoietic stem cells, there is the immune reconstitution with a more tolerant immune system. And this eventually will lead to very, very significant results. And I'm just quoting briefly data produced by several groups that have been summarized by Paolo Muraro just a few years ago, showing that indeed, this treatment can significantly impact the disease progression, particularly in patients with relapsing remitting MS and with low EDSS. And while in progressive MS, there is still an effect because you can see here the steepness of the curve is pretty, is quite steep. While here you have kind of a decrease in the steepness, but however, it is rather obvious that as for any other treatment, as suggested by Professor Comey, early phases in inflammatory diseases are the best target for any treatment in MS, but particularly for transplantation. This is a recent study published by the Canadian group showing indeed a very dramatic effect on MS activity, relapses, and MRI activity upon a rather strong myeloablative conditioning, meaning the immunosuppressive phase of the treatment. And just to give you an idea, just to provide you with what have been shown to you by Professor Comey, this is the level of NIDA reported at 2 years for the current studies published in the literature for hematopoietic stem cell transplantation. As you can see, there is a rather evident difference, although a phase 3 trial has never been performed due to rather clear and obvious reasons. It is, I would say now, pretty much accepted also from those that were skeptical about this treatment that this is an extremely effective treatment for patients with multiple sclerosis. Now I move to my specific field, which is mesenchymal stem cells, and I will try to explain you why a few years ago, actually at this point about 15 years ago, we decided to move on, and thanks to the support of many people, but as I said, particularly the Italian MS Society, we move into a trial that is now close to the end. So mesenchymal stem cells are cells that by default are expected to produce bone, fat, and cartilage. So why these cells should be utilized to treat multiple sclerosis? Not only where do you get these cells? Are they in the central nervous system? Not at all. They can be isolated from almost any connective tissue, but those that we and most of the people use are those from the bone marrow, where they closely interact with hematopoietic stem cells, and they play a major role in the maintenance of the hematopoiesis. Now, as I said to you, why we should use stem cells that are in principle able to produce fat, bone, and cartilage to treat multiple sclerosis and eventually other neurodegenerative disorders. Now, the reason to think of that came from a paper that was published in Science in 2000 showing in a very attractive way. I think that this study was particularly interesting. I mean, it was not particularly interesting, and the data were not confirmed at all, but was really sexy because the title was Turning Blood into Brain. It's not even true because MSCs are not blood cells, are stromal cells. However, this title moved into Time, into Newsweek. It was published in all the major papers because, come on, I mean, turning blood into brain. And the reason is that eventually some people have been able to demonstrate that under certain circumstances, mesenchymal stem cells can acquire, rarely, sorry, I made a mistake, is an A, rarely, they can transdifferentiate into cells that have a phenotype of neurons, which doesn't mean they are neurons. However, now, tons of people have demonstrated that MSCs can protect neurons when you induce some level of toxic damage to those cells, in vitro and, as you will see, in vivo. More interesting, although I have to say that these are actually just animal data, have been, now many others have shown, but I like to present these early studies in which it was demonstrated that if you inject mesenchymal stem cells into mice with a certain lesion in the central nervous system, you can enable a reactivation of those cells in the central nervous system, the neural precursor cells that Gianvito and Stefano Pluchino and Laura Ottoboni have been showing to be very important in the modulation of immune response, in tissue protection, and even in tissue repair in the central nervous system. So these MSCs injected can reactivate some path, intracellular path, endogenous path that may lead, at least in mice, somehow to tissue repair. So what we call endogenous neurogenesis, eventually promoted by mesenchymal stem cells. Of course, I have an immunological background, and the reason because I started to think of MSCs was because we and others have demonstrated that mesenchymal stem cells can inhibit several types of cells of the immune system, particularly T cells and B cells that we heard from Professor Comey playing a major role in the pathogenesis of multiple sclerosis. So the idea that I had, and was my first submission to a grant to the Italian MS Society in 1992, that if we had cells that eventually could become neurons, and at the same time they can inhibit T cells that are playing a major role in damaging those neurons, we can have a single cell that can do a dual job. So that was the idea, very simple, very naive, but I would say quite innovative. And I said a few times during conferences that 10 years later, I heard that Hartmut Weckerle was the reviewer of that project and was very encouraging and very positive to that grant that actually was funded and led to the first demonstration that if you induce EAE and you inject mice with mesenchymal stem cells intravenously, you can ameliorate disease. So what? Many other things have done that. But you can, for the first time, we were able to show that if you isolate T cells or B cells from these mice and you stimulate with myelin antigens, self-antigens, these cells were not responding anymore. So we were able to, at that time, to induce immune tolerance. Now, okay, most of the people here are professionals or people with MS or somehow stakeholders, and probably the answer would say, okay, who cares? We have a lot of great drugs, as Professor Comey showed us, is what we want from stem cells to be able to do immunomodulation. So we attempted to demonstrate whether they were able to transdifferentiate. And unfortunately, we could not observe almost any level of engraftment. So if you inject stem cells intravenously, those cells do not get into the central nervous system, or at least to a very limited extension. Even if you inject them intracisternally, so what we could do in patients with MS, injecting intrathecally, we could not observe any level of engraftment. However, when we measure the amount of axons, neurons, in the spinal cord of these mice, we could observe about 3 times the number of axons, suggesting that somehow we were able to induce some neuroprotection. Now, what are the causes of tissue damage in MS? There are several. I'm just picking up some data from all the story that we produce, all the science, the little science we produce. And one of the major cells producing damage in MS and in many other diseases are microglia. We heard from Professor Comey. Now we demonstrated that MSCs can inhibit those factors released by microglial cells, by macrophage, that can contribute to damage and to tissue derangement. The cells that produce toxic factors such as pro-inflammatory cytokines or derivative of nitric oxide are, as I said, microglia, and we demonstrated that mesenchymal stem cells can change the phenotype of microglia in vitro and later in vivo. But what I wanted to make toward a neuroprotective phenotype. But what I wanted to, the point I kept this slide for was that this was mediated by a molecule that was released by mesenchymal stem cells, and this was the first demonstration, suggesting that a concept that now is very obvious for most of the people in the field, that stem cells can release factors. They do not necessarily have to get there or get in touch with other cells to change, to have an impact on the disease. In this case, was the chemokine was fractalkine, and we demonstrated that if you inhibit fractalkine in mesenchymal stem cells, you cannot have any more this effect on microglia, but this doesn't matter. The point was that mesenchymal stem cells can interact with other cells through the release of factors. So now these are new data. I decided to provide you with, I'm not going to go through the details of the experiments. Most of you, or many of you, are not directly scientists, but I wanted to make a point to show you with new stuff that things are really moving fast, hopefully providing some more info, more clue toward the possible application of this treatment also for people for progressive MS or with degenerative diseases. So in this case, together with Claudio Verderio in Milan, we expose microglia to MSCs, and as we know, Claudio Verderio and Roberto Furlan from San Raffaele have demonstrated that these microglia cells can release microvesicles. And guess what? Microvesicles released by MSCs exposed microglia can enhance repair of myelin in mice with focal lesion of the spinal cord. And we also could observe that these microvesicles released by MSCs exposed microglia can enhance the proliferation of oligodendrocyte precursor cells, which are the cells that produce myelin. And indeed, when we measure the amount of myelin produced by OPCs exposed to these microvesicles, we could observe that this is indeed was the case. So very promising, very preliminary, very animal data, but very promising to us. Another bunch of new data suggesting how we need to imagine MSCs could work in, hopefully in MS. It has been recently demonstrated in a Nature paper that was really a pivotal study by Ben Barres that astrocytes, another cell type in the central nervous system, can acquire a detrimental phenotype that kills neurons. Now, we took those astrocytes and we tried to see upon inflammatory cues whether or not in the presence of MSCs their phenotype changed. And indeed, this neurotoxic phenotype seemed to be inhibited, these are yet preliminary data, seem to be inhibited by MSC-derived exosome at the molecular level and also the protein level, as you can see from this beautiful confocal data showing that markers or cytokines produced by detrimental or neurotoxic astrocytes can be inhibited by MSCs, by the interactions with vesicles derived from MSCs. Another thing that astrocytes can contribute to is to the development of EAE and the damage in EAE toward inhibition or remyelination. These molecules, called endothelin-1, is a supposed factor inhibiting remyelination or recruiting through CCL2 the influx of other cells in the central nervous system. And MSCs inhibit that both in vitro and in vivo. Another piece of appealing science is the possibility that local or endogenous repair could be promoted, not only recruiting NPCs, neuroprecursor cells, but also astrocytes. A certain type of astrocytes can acquire a stem cell-like phenotype. And Tiziani in the lab were able to show that in the presence of interferon gamma, which is an inflammatory cytokine associated with multiple sclerosis, MSCs can induce the formation of astrospheres, so they induce astrocytes to become cells with the potential of stem cells, and indeed these cells have a kind of a neuronal and astrocytic phenotype. So it seems that MSCs can tell astrocytes, go back to a more stem cell phenotype and eventually differentiate into something new, including neurons that can help possibly the tissue repair. Recently, we've been using, together with Alessia Cedola here from CNR and in Grenoble, the synchrotron, we use a new technique called the synchrotron X-ray phase contrast tomography to address in a very unique and detailed way the three-dimensional anatomy of structures in the central nervous system. You can see we can even detect with a microscopic detail the phenotype, the morphological phenotype of motor neurons. Now, we use this technique to see whether or not MSCs have an impact on capillaries. Now there is a very interesting new interest in the field about the role of vessels, microvessels, and vascularization in the development of EAE and multiple sclerosis. And as you can see here, MSC-treated animals with EAE have a much higher number of capillaries compared to EAE animals, control animals. And we could also observe that if you consider that these are motor neurons, this is a video, but I'm not going to start it, in animals with EAE, there is indeed, and you can see better in this slide here, these are motor neurons. This is a normal animal. These are animals with EAE. You see a significant loss of motor neurons. And in this case, you can see that upon MSC administration, these motor neurons are preserved in the spinal cord of these mice. What about the blood-brain barrier, we show that MSCs can, again using this technique, significantly inhibit the extravasation, actually the extravasation of fluids, I'm not talking about cells, upon administration intravenous in mice with EAE. These are control mice. You can see clearly here a cloud of exudate, and this is significantly inhibited. And we've been showing together with Daniela Virgintino in Bari that this is probably through the ability of mesenchymal stem cells to restore the integrity of the blood-brain barrier. And as you can see, this is potentially, again, these are mouse data, so I'm not selling here anything to say that this is going to be the case for MS, but it's really appealing to hope that this could be the case. And I wanted to just show you some not MS data, because this is important for progressive patients with MS, because people at the beginning of this story were telling us, okay, all this is due to immunomodulation. Indeed, in a model of stroke, in a model of ALS, in a model of epilepsy, and these are just few examples, but these are produced from us, we could observe a significant impact of MSCs in neuron preservation, survival of mice with ALS, upon intravenous MSCs administration in animal models in which adaptive immunity plays a very minor role. So that means lymphocytes. And just to give you a last flavor of what we have been doing, this is a double transgenic mouse model of Alzheimer. We are using MSCs which have been conditioned with Alzheimer extract, meaning Gianvito have been pushing, and I'm fully in agreement with him, the concept of plasticity of stem cells. What means plasticity? It means that a cell can change his behavior, his functional behavior, depending on the environment. Meaning, if I sense inflammation, I'll do something different if I sense necrosis. Now, in this case, which is indeed the best part about using stem cells, because, I mean, you believe that they are somehow intelligent rockets. Now, in this mouse model, we use MSCs that have been exposed to Alzheimer extract, and this makes these cells much more efficient. This has been done in collaboration with Claudia Balducci and Forloni at Mario Negri, and we show that upon intranasal, also intravenous, but intranasal administration of the supernatant of these MSCs, we can significantly ameliorate memory in mice, and we can better clear amyloid plaques in this mouse model. So this is a bit of my view of how MSCs could work. MSCs certainly, and I didn't go through those old data, because you may have heard that already several times, they can inhibit T cells and B cells producing toxic factors or antibodies or cytokines. They can change the phenotype of macrophage and microglia that are favoring or damaging axons as well. They can directly protect neurons. They can inhibit astrogliosis, detrimental astrogliosis, eventually inducing the change of a phenotype through what has been called a non-cell autonomous mechanism. Cell autonomous means these cells act directly on the target cells. Non-cell autonomous means that there may be a second or third or fourth cell that play a role, so MSCs may impact neurons' life, not necessarily directly, but through astrocytes, through microglia, through interactions with immune cells. Now, finally, Gianvito convinced me, so please tell him that I put this quotation of his paper, that indeed he was the first one to show or to provide this concept. We don't need necessarily cells that engraft in the central nervous system. We don't need to necessarily have cells that become neurons to protect and possibly favor tissue repair. Now let's move the last 5 minutes to mention where we are with the trial that have been supported by many agencies, mainly the Italian MS Society, but also the MSIF was kind enough to support us at the beginning and in this effort. And the concept was rather simple. We first, but still unique in the field of science, because rarely, although things are changing and Paul has been pivotal, playing a role in trying to put together scientists that usually fight among them, but this was in 2010, and we met in Paris, and instead of saying, look, why we keep going with single studies with 10 patients? We will not go anywhere, we will not provide any science, and we will still have patients going to whatever island in the middle of the ocean to get stem cells to be treated. So we agree on a protocol based on the preclinical data that I provided to you, and we develop a study, which is a double-blind phase 2 clinical trial, which involved 12 nations. Italy has played a pivotal role, together with Genova, also San Raffaele Hospital in Milan and Verona Hospital have been involved. This is the trial design, meaning that patients have been exposed to MSCs either at the time 0 or were administered 6 months later. So each patient will be, or have been, because now we finished the enrollment and the last patient has been treated, as you will see. So each patient has been exposed to MSCs either at time 0 or after 6 months. The primary outcome was safety, because still we are talking about an experimental treatment, but also the ability of MSCs to have an impact on what has been so far the most accepted, and I would say easy, but you can imagine this is not an easy trial, easy target to demonstrate that MSCs are biologically active. However, we are very much interested in all the secondary objectives. There are those that will help us to at least smell a flavor of neuroprotection and neurorepair and tissue repair. This is where we are. We have been screening, we screened 163 patients. 144 have received at least one infusion. We have 120 patients that have been that completed the study. 4 are still ongoing. We are expected to lock the database in January 2018, and we hope to be able to present data to the American Academy as late breaking news if this will be accepted, but we are running to get those data for. And then, of course, present the full body of data for the ECTRIMS 2019. This is the screening failure, 19 patients. In most of the cases, we have patients whose cells fail to grow. In some cases, 2 cases, we had disease progression. Disease progression, of course, because you need a certain time by the screening, and then you think that at a certain point the patients could receive placebo. There is the possibility that for a patient that for 8 months will be in placebo. So when we started this study, most of the drugs are available now were not available. So in some cases, in the most recent years, we decided to withdraw the patient. The good thing is that the total serious adverse events were limited, and they were never related to study treatment or procedure by an international data safety monitoring board. And this is my personal view of what I think MSCs are. Is a cargo cell that is loaded by goodies that can be eventually delivered depending on cues that are associated to different diseases. Now, because Gianvito is not here, I will just provide you in 2 minutes some I wouldn't say data because I'm not able to present Gianvito's data, but just to tell you that it has been done an enormous work by the San Raffaele group and Gianvito, and the basis is quite complex, although I would say that somehow there are the mechanistic pathways are different, but conceptually we can say that many adult stem cells work in a similar way, meaning also neuroprecursor cells do immunomodulation. Also, neuroprecursor cells can protect neurons and through bystander mechanisms can completely or significantly change the pathological environment that occurs in EA. It is obvious that the potentiality of neuroprecursor cells to transdifferentiate or to engraft in the CNS is certainly superior to MSCs because they are neural cells. However, Gianvito, I imagine, I heard him several times, would say that probably cell replacement is not the major mechanism for this drug. Where are they? They are doing pretty good as well, although technically MSCs are much easier, so it took much more time for them to go through a roadmap than move to the lab, to the clinic. But however, finally, San Raffaele was able to start about 1 year ago a phase 1, which is mainly addressing the safety of this treatment on patients with the progressive disease, with an EDSS pretty high, as no other options, therapeutic options could have been possible at the time of inclusion. And there is no clinical data as it is expected because the study is ongoing, and in any case is a phase 1 study, but that's just that to show you that treatment with an NPCs now is a reality, and future studies beyond this one will help us to say whether or not there is any clinical effect. However, being part of the data monitoring board, I can confirm that any safety-related issue is not present in this trial so far. So very appealing as well. So that's all, and I just wanted to thank for those data that were produced in my lab and collaborators, many people that contributed a long time, and of course the Italian MS Foundation for supporting me in all these years. Thank you.